In the last video, we went over a general overview of the mechanism of DNA replication and talked about some of the components. I would strongly recommend that you, before continuing this video, you go back into Blackboard and look for the DNA replication video. It's under a tab that says something like videos from another textbook. This is a really good animation of what is happening. This process is a moving process. There are a lot of parts, and it doesn't really make sense on paper to show it sort of two-dimensional. It's really better in 3D with all the moving parts. So I'd suggest that you watch that. There are a couple different videos in our chapter 4 information on Blackboard. And once you watch it, come back, we'll talk about the specific parts, and then I'd suggest watching it again just to get a good overview. So you should see this Bioflix DNA replication. I think the file is called something like 13-20 DNA replication. It's just a really good video from another textbook that I've used in the past. So that's there on Blackboard if you want to go watch that. So although the idea of DNA replication is really simple, this idea of semi-conservative using each of the parent strands as the daughter strands template is really simple, but there are actually a few complex bits and pieces and challenges that are encountered when we start talking about replication. So we're going to go through each of these challenges, starting with how does polymerase actually get started? So remember that DNA is made by adding nucleotides to the 3' hydroxyl group. But that requires a free, it actually requires a second strand. It requires that complementary strand is already started. And so you need to have that free 3' end. Now, one thing that DNA polymerase cannot do is that DNA polymerase cannot add to a strand without a nucleotide to add to. So if we look at this part down here, this bottom right hand part, we have a free 3' end that we can add it to. But if you look back here, this very first thymine here, if you pretend that this sequence isn't there, you'll notice that there's not actually a free 3' end to add to, and that's because this guanine over here is still tied up within the replication fork. Okay, so that's a problem. Up here, we have a free 3' end, so we can add nucleotides. But when we were back in our replication bubble with nothing going on, and we needed to start replication, we didn't have another nucleotide here to add to. We have essentially a naked DNA strand. And so to overcome that problem, we need to add some free 3' ends. And we accomplish this by adding what is called an RNA primer. Now, this is going to be analogous to that PCR primer that I told you about, where we essentially start adding little nucleotides in there. The PCR primer comes as a packet, but our RNA primer is going to be made up of little nucleotides that are added without that 3' end. So, oops, I went too far. So the first thing we're going to do then is actually bring in another enzyme called primase. So primase is an enzyme that can add RNA molecules to the DNA strand without that free 3' end. Now we'll see a similar molecule when we get to transcription in RNA polymerase. So RNA polymerase is another molecule that can add RNA without having that free 3' end. So this primase molecule is actually going to add a short segment of RNA. And we know this is RNA because we have uracil in there. But the reason for this is that it produces that free 3' end to which we can attach the incoming nucleotide. Now, keep in mind that we're going to have this RNA mixed in with our DNA, so we're going to have to deal with that later. But to get us started, that RNA primer is going to come in and, the RNA primase is going to come in and start adding those nucleotides so we can get our DNA replication started. So our next problem is the polarity. Remember that these strands are anti-parallel. So that means that one strand goes in 5' to 3' direction. The other strand runs 5' to 3' in the opposite direction. Now, because DNA chains are extended only at the 3' end, we're actually going to have to do replication in 2 different directions. And this is because of the location of those 3' ends. So on one strand, we're going to need to move polymerase one direction, and on the other strand, we have to move a polymerase molecule in a different direction. At the same time, we have our replication fork expanding outward. So if this is our analogous location here, helicase is going to continue moving and expanding our replication fork. So how are we going to replicate both strands at the same time in opposite directions? Right, so we can do this by replicating them slightly differently on each strand. Now on one strand, we are going to produce a continuous strand. So our polymerase is going to bind. It's going to move in a 5' to 3' direction towards our replication fork. And we can do this continuously because once we have that RNA primer that was out here ahead of all of this business, once we have that RNA primer, we can just continue in the 5' to 3' direction. And we're going to follow that helicase molecule as it moves down. So this strand that is produced continuously is called the leading strand. On the opposite strand, and at the same time, we have to do things a little bit differently because that polymerase is moving in a different direction. So on one strand, we have continuous replication, and on the other one, we have to do things a little bit differently. So at this replication fork, there's only so much space where our RNA primer can get into. So even if the RNA primer comes in at the farthest nucleotide within that replication fork, we still are not replicating anything farther down. So what happens is as this replication fork moves in, RNA moves along. RNA primer comes in and it binds as soon as it can in that replication fork, and it makes a little RNA primer. And then a DNA polymerase molecule comes in, binds there, and continues adding onto our strand. Now, as that helicase moves along the replication fork, here's our helicase again, as that helicase moves, it's going to open up more exposed strands. And so another RNA primer can come in and lay down, and a second DNA polymerase can move in and continue working this way. Now, you might notice that we're starting to see fragments here. So eventually, as your strand opens up, we are going to, on the other strand, have this series of fragments called Okazaki fragments. And this strand with the fragments is called the lagging strand. Now the lagging strand is made discontinuously because as the fork opens up, we can now make another part. So on one strand, we're going to be working continuously, moving toward the replication fork in a 5' to 3' direction. On the other strand, we're going to be working discontinuously, still in a 5' to 3' direction, but we have to make those fragments until we get farther, or because we keep moving farther along that replication fork. So I like this diagram because it removes some of our complex pieces, but up on top we have that lagging strand. That's the one that's going to be working in a continuous motion, and that's going to just continue all the way along following that replication fork and the helicase molecule all the way down. So on the lagging strand, we can see now that we have these fragments, and we'll continue to produce fragments as long as that leading strand and helicase are opening up that fork. So all of these Okazaki fragments now are going to continue their extension until they bump into the next fragment on the line. But these are our RNA primers here, the little purple spots. This is RNA. Now we're making a DNA molecule, so we need to get rid of that RNA. You cannot connect DNA to RNA, and at the 5' end, you cannot add or create a bond from this direction. So we actually need to remove our RNA, and then we need to fill that little gap from the RNA with DNA. So a second enzyme is going to come in once polymerase has moved on, remove that RNA. I believe this is RNA polymerase 1. I'm sorry, DNA polymerase 1. DNA Pol 1 is going to come in here, and it's going to remove the RNA, fill it in with DNA, and then DNA ligase is going to come in and close these little gaps. It essentially is going to link all of the DNA together that was filled in when we removed those RNA primers. And so you end up with your leading strand, doesn't have any of those primers. It had one way back here at the origin of replication, but you only need to do it at that one spot. On the other strand, we're going to fill in all that we need. Now I want to mention here that remember that this is a bubble, and so you have the same thing happening on the other side. And in fact, if you start at the origin and you make a leading strand in a, sorry, this is backwards, 3', 5', in a 5' to 3' direction, you get your leading strand on the top. And the bottom is still a 5' to 3' direction that we're making on that new strand that we're building. Still a 5' to 3', but we've got our little fragments. Now, if you work this out, if you look at what's happening, we also have another a fork, another replication fork here. So here we are actually going to have the opposite of what's happening on that top strand. So here we're going to have DNA polymerase bind here and move toward in a 5' to 3' direction, adding on to the new strand. At the replication fork, we're going to have our fragments again, because here is our 5' end, 3', and our little fragments. I know that's getting kind of messy. But you can see this is sort of indicated if you go back a couple of slides to our original slide showing the replication bubble. You can see what's happening, and actually I'll go ahead and do that for you. So here's our replication bubble that I was talking about. We've got our origin here, and we'll move in a 5' to 3' direction there. And then our fragments on the other strand. And then over here, we move in a 5' to 3'. Now, this all goes back to this anti-parallel problem and the fact that we have to produce anti-parallel strands. So this idea of having Okazaki fragments on both sides of your replication bubble tends to confuse students. You can unconfuse yourself by drawing your own bubble, even pulling it apart completely and just looking at your strands and figuring out where you're going to have your anti-parallel strands. And if you mark the origins like I have here, you can look at which directions your 5' to 3' are going to have to go in. I would strongly recommend you drawing that. I like to ask that question, ask students to figure out which is the leading and which is the lagging strand. If you have questions, let me know and we can go over this in lab. The other thing I want to point out is that we tend to draw these strands in the same direction every time, but I might give you a strand where the 3' is actually on the top. So be able to look at it in both directions and figure out which one is your leading and your lagging. All right, so challenge number 3, the unwinding problem. Now remember, if you have tried this rubber band experiment, you will see the tangling that I was talking about. So remember, if you have a rubber band, you can twist it up and attempt to make a replication bubble. And if you continue to separate this bubble and make it wider, you're going to get really tight here on the ends, and that's going to break your DNA. So without those topoisomerase enzymes, you are actually going to supercoil your DNA down here and end up breaking your DNA. So topoisomerases come in and actually make a single strand break and then allow the molecule to rotate around the other strand to release some of that tension and then seal it back together. So this is really important because that helicase is putting pressure on this molecule down here because it's attempting to pull apart that replication bubble. So topoisomerases are really important in keeping the damage to the DNA at a minimum. All right, challenge number 4, replicating the